and welcome to today's STERIS Tech Talk on reusable device testing. STERIS Tech Talks are a series of webinars covering subjects relating to gas and radiation sterilization processing and the laboratory testing and validation services which support these processes. My name is Ashley Marute, and I'm the Associate Product Manager for STERIS Applied Sterilization Technologies. I'll be the host for today's event. Our presenter today is Tim Skordahl, a project manager for reusable devices. Tim has served as a project manager at STERIS since May of 2015 in the Industrial Validations Group with a focus on steam sterilization and reusable devices, and is currently the subject matter expert for moist heat and dry heat sterilization, and also for processing of reusable medical devices. Tim provides technical and sales support for new projects, working closely with customers, account managers, and other groups across multiple business segments within the STERIS organization to answer questions, provide cost and timeline estimates, and to ensure resources are available to support new projects. Tim is involved with AAMI and ASTM standards organizations and has over 20 years of experience with process validations and medical device manufacturing and sterilization, having served in various roles at Viromed Laboratories, Laboratory Corporation of America, Biotest Laboratories, and the last 10 years here with STERIS. All attendees are on mute for the presentation. However, we would like to encourage everyone to submit questions using the questions function on the GoToWebinar control panel. Questions will be answered following the presentation. Today's presentation will be recorded and uploaded to our STERIS Applied Sterilization Technologies YouTube channel. Please note that continuing education credits are not provided as part of this webinar. And now over to Tim to begin the presentation.
Good morning, good afternoon, or good evening, whichever the case may be. I am really delighted to be here presenting to you, and I hope each of you enjoy this little presentation as we walk through it. For the agenda today, I'm going to give you a little bit of a background on the reusable device industry. We're going to review the processing cycle, what used to be called the reprocessing cycle. We're going to spend a bit of time discussing cleaning process validations and disinfection process validations, sterilization process validations, and then really quickly, I'm going to present a slide on looking ahead with regard to some standards that are coming down, will be published very soon. So when we talk about medical devices, there's two high-level categories of medical devices. There's a single-use device and a multiple-use device, which is a reusable. So single-use medical devices are labeled or intended to be used on one individual during a single procedure. Reusable medical devices are devices that are designated or intended by the manufacturer as suitable for processing and reuse. Now, FDA in their industry guidance has told us that their industry guidance is applicable to reusable medical devices. They have defined four categories that they consider to be applicable to their industry guidance. The first three are very familiar descriptions of reusable devices. Number one is a device that's initially supplied sterile and requires the user to sterilize it, clean and sterilize it after the initial use. The second category is a device that's initially supplied non-sterile and requires the user to clean and sterilize before the initial use. And then the third category is a device that is intended to be reused by a single patient but processed between each use. But the fourth category, which sometimes gets overlooked, is actually a single-use medical device that is initially supplied as non-sterile to the user and requires the user to process the device, which means clean and sterilize it, prior to its one and only use. So when you're in this fourth category, your device gets treated essentially exactly like a reusable medical device, even though technically it is a single-use device. There are four primary standards that we use in the reusable device industry. There are some others that are used, but these are the four ones that are used primarily from a manufacturer, a device manufacturer's perspective. There's ANSI AAMI ISO 17664, which is an international guidance document published in 2017, processing of healthcare products. information to be provided by the medical device manufacturer for the processing of medical devices. And then domestically, FDA has their industry guidance, which was published in 2015 and then updated in 2017. I think they tweaked one of the annexes or something. It wasn't a major change in 2017. And that's titled Reprocessing Medical Devices in Healthcare Settings, Validation Methods and Labeling, a Guidance for Industry and Food and Drug Administration Staff. And then there's AMI TIR 30, which was published in 2011, a compendium of processes, materials, test methods, and acceptance criteria for cleaning reusable medical devices. And AMI TIR 12, which was published in 2010, and that's titled Designing, Testing, and Labeling Reusable Medical Devices for Reprocessing in Healthcare Facilities, a Guide for Medical Device Manufacturers. So let's take a look at this processing cycle. For those of you who have sat in on some of my other presentations, you've seen this diagram before. I'm going to walk through this really quickly so we can get a better view, item by item, how this process works. So we start with a brand new device. This device will usually enter the processing cycle at the cleaning stage. After it's cleaned, there's an optional interim disinfection stage that may or may not be, it just depends on how your IFU is worded, on the use of your device. It's kind of an optional process, but it's called interim disinfection. After that, the devices are inspected and packaged. If during the inspection process, a device is found to be broken, it needs repair, that device can either be repaired or discarded. If it is repaired, it returns to the process back up here at the cleaning phase and goes back around the circle again. After inspection and packaging, we have either sterilization or terminal disinfection, one or the other. They're both microbicidal processes, depending on the device capabilities. If it cannot be sterilized, then basically the default is to apply a terminal disinfection process to it. After the microbicidal process is performed, they are put into storage. Devices that are sterilized typically have a sterile barrier system, so they are usually put into a larger room on wire racks with good ventilation, environmentally controlled, just a nice clean room. Items that are processed with a thermal disinfection process typically are not going to have any kind of a sterile barrier system. So these will be stored in smaller, environmentally controlled cabinets to make sure that they don't get reinfected prior to patient use. So after they're stored for a period of time, they go to patient use. After the procedure is done, they re-enter the cycle again back at the cleaning phase. And then we go around this circle, and that's how a reusable device functions from beginning. Excuse me. Let's look at a couple of definitions really quick. The definition of the word processing can be found in ISO 17664. It's an activity to prepare a new or used healthcare product for its intended use. And then it goes on to say that for the purposes of this document, processing includes cleaning, disinfection, and sterilization, if necessary and applicable. Cleaning is defined in ISO 11139 as the removal of contaminants to the extent necessary for further processing or for intended use. AMI TIR 30 defines disinfection as a process that kills pathogenic and other organisms by physical or chemical means. And AMI TIR 12 defines sterilization as a validated process used to render product free of viable microorganisms. So regarding validation of the processing cycle, ISO 17664 tells us that a medical device manufacturer shall validate each process that is identified in the information supplied with the device. So that's your IFU. Any process that is defined in your IFU must be validated. They also say that the medical device manufacturer must have objective evidence available that the validation was performed, and that's going to be your final report that resulted from the validation. And then it goes on that at least one validated method shall be specified for each applicable stage of processing of the medical device. The FDA industry guidance says that cleaning and disinfection of sterilization processes should be validated to provide a high degree of assurance that the device will consistently meet predetermined specifications. And then they also say that cleaning, disinfection, and sterilization methods should be validated, and the validation should be completed prior to submission. The FDA industry guidance also says that a method of cleaning, your labeling should provide a detailed, validated method of cleaning. And then for a method of disinfection or sterilization, it says that for reusable devices intended to be disinfected or sterilized, reprocessing instructions should specify at least one validated microbicidal method of disinfection or sterilization. AAMI TIR 12 says very similar things. It says you have to specify a validated method for manual cleaning. There's texting here about high-level disinfection, make sure that the devices can be high-level disinfected. They also talk about supporting label claims for sterilization to be validated. And then finally, TIR 12 also talks about if storage or transportation conditions could affect the safety, functionality, or useful life of the device, the manufacturer should recommend an acceptable range of storage and transport conditions. This is essentially a shelf life validation. So we're back to this image again. Now that we've walked through a little bit of what the standards and the FDA industry guidance tell us what needs to be validated, let's define, according to this cycle, what needs to be validated. So obviously, yes, cleaning needs to be validated. If your IFU defines an interim disinfection process, so for instance, if you clean your device in an automated washer disinfector and your IFU tells your users that they are to use a disinfection process with the water at a certain temperature for a certain amount of time, that process is your interim disinfection process, and that needs to be validated. Your sterilization or terminal disinfection process certainly needs to be validated. And under certain conditions, your storage, your shelf life needs to be validated. One item that's often overlooked with regard to validation of the processing cycle is the entire cycle itself. How many times can your device be processed without suffering degradation to the point to where it can't be used anymore? So TIR 12 tells us that if the device is adversely affected by the cleaning, disinfection, or sterilization process, the limits of reuse should be determined. FDA industry tells us that labeling should either inform the user how many times the device can be reused or provide the user with a mechanism or method to ascertain whether the device has exceeded its use of life. So let's take a closer look at cleaning processes. We're going to walk through cleaning, disinfection, and sterilization separately. For the cleaning process, we'll go a little bit deeper into the definitions. ISO 11139 and AAMI, ANSI AAMI ISO 17664 have the same definition. It's the removal of contaminants to the extent necessary for further processing or for intended use. So there are some devices, some medical devices, that really only need to be cleaned, and then they can move on and be reused again. TIR 30 and TIR 12 share this following definition. Cleaning is the removal of contamination from an item to the extent necessary for further processing or for the intended use. Very similar verbiage as to what ISO says. And then the FDA industry guidance defines cleaning as the physical removal of soil and contaminants. Later on, they say cleaning is the first step in the reprocessing and should be described in the labeling as part of the overall reprocessing instructions. TIR 12 tells us that validation of the decontamination procedure should separate cleaning and disinfection. So if you're putting a device, if your IFU recommends an automated cleaning process in a washer disinfector, you need to validate the cleaning aspects of that process separate from the disinfection aspects of that one process. So yes, you're putting it into one machine and you're pushing one button, but you're performing two functions in that machine, and both functions need to be validated separately. So TAR 12 goes on to say that specifically a validated method of manual, or specify a validated method of manual cleaning, and at least one method of validated automated cleaning unless your device can't withstand an automated cleaning. FDA industry guidance says simply that the labeler should provide a detailed validated method of cleaning. They don't define whether it needs to be manual or automated. So with regard to cleaning processes, there are essentially three categories of cleaning processes. So we have either a manual cleaning process, what I call a semi-automated cleaning process. I don't know if that's a widely used term. I haven't thought of a better one yet or read a better one yet. And then finally, there's an automated cleaning process. So the manual cleaning is cleaning without the use of any kind of automated equipment. Semi-automated cleaning is manual cleaning with the added use followed by sonication or cleaning in an ultrasonic washer. And then a fully automated process is simply using a washer that's compliant with ISO 15883 for a fully automated cleaning process. In addition to the three categories of cleaning processes, we have to look at detergents when we're validating a process. So in the Western Hemisphere, we generally prefer a pH neutral enzymatic detergent. That's just a high level classification of the types of detergent that the Western Hemisphere prefers, the US prefers. In Europe and in Asia, they actually prefer an alkaline pH, non-enzymatic detergent. So each unique combination of processing categories, which is manual, semi-automated, and automated, and then each detergent classification, they need to be combined and validated separately. So each unique combination needs to be validated separately. So for instance, if your IFU says that you can manually clean your device with a neutral pH enzymatic detergent, and you can put your device through an automated washer disinfector using a neutral pH enzymatic detergent, or you can use the same washer disinfector process with an alkaline pH non-enzymatic cleaning chemistry, then you're looking at three separate process validations because there are three different unique combinations of processes, and they each should be validated separately and uniquely. Now let's take a closer look at how we validate a cleaning process. So we generally start with 6 replicate devices. 3 of those devices are going to be control devices. 3 of the devices will be test devices. Those 6 devices go through what we call a simulated use process. So what happens is the test devices will get soiled, the control devices do not get soiled, but then all 6 devices get cleaned. And then we repeat that cycle a total of 6 times. And we do that for 2 reasons. First, we don't want to perform a cleaning process validation on a smooth, pristine, brand new device that doesn't have any, micro fractures or scratches or, just different things that happen to a device over use. Secondly, we want to allow for the potential of residual soil to build up on the device over multiple uses. So if you have a brand new device right off the shelf and you wash it and clean it one time, and then if you were to test it for residual soil, you will likely fall below the method of detection. So you'll say, yay, my cleaning process works. I have nothing detectable. But then if you wash that same device 6 or 7 or 8 or a dozen times, you might slowly build up soil each time to the point to where if after 6 uses, or in this case 7 uses, you might actually wind up with a level of soil on your device that's detectable by our assays. So after we go through the simulated use, we soil the test devices again a 7th time, and then we'll clean them a 7th time. And then we extract the negative sample controls and the negative device controls and the test devices. So we have 3 sample sets, a negative sample and negative device control, as well as the test devices. And then those get, after they're extracted, we do analytical testing on the extract fluid. We typically test for TOC and protein. These are the 2 assays that STERIS recommends. I know that hemoglobin is a very popular marker that's used for cleaning process validations. I personally don't like the combination of protein and hemoglobin because you're essentially testing for the first thing. Anything that spikes a hemoglobin test is also going to spike a protein test. So I prefer using TOC and protein, and I think overall in the industry that's becoming a widely accepted combination. All of the standards, by the way, do require 2 markers, not just one. So that's why we test both for TOC and protein. So after we've tested the negative controls and the test devices, we'll repurpose the negative device controls as positive device controls, and then we will soil them with the same test soil. We'll extract the positive sample controls and do the analytical testing, the same TOC and protein, for positive sample controls and positive device controls. And then using the values from the positive sample controls and the positive device controls, we're able to determine a recovery efficiency, or how much of that soil are we actually able to get. So if we know, if we put, for instance, if we know we put 5 micrograms of soil on your test device, and I'm only able to recover 4 micrograms, then we know that our recovery efficiency is only 80%, and we have to have a correction factor of 20% added to whatever we recover from our devices. So we determine the recovery efficiency, and then that recovery efficiency is applied to this testing that was done previously on the test devices to show whether or not the test devices were actually able to be effectively cleaned and met the acceptance criteria for both TOC and protein. After we're done with this testing, after we've done validated the cleaning process, we will take the same 3 test devices again, and we'll actually go through back-to-back 2x cleaning using a maximal cleaning process. I should back up a little bit and say that up here, when we clean the test devices during simulated use, we use what we call a nominal cleaning device, or I'm sorry, nominal cleaning process. And then for this cleaning process right here, this one time, we'll use a minimal cleaning process where we're using shortest durations, lowest chemistry concentrations, that sort of thing. And then down here, we'll use a maximal cleaning process as a worst case, because we're going to be doing detergent residual testing. So we'll clean the test devices 2x using the maximal cleaning process. And then we do cytotoxicity testing to test for any residual cleaning chemistry that might still be on the device. So let's talk a little bit about disinfection. With the disinfection process, ISO 11139 and 17664 define disinfection as a process to inactivate viable microorganisms to a level previously specified as being appropriate for a defined purpose. AAMI TIR 30 and TIR 12 both share this definition: a process that kills pathogenic and other microorganisms by physical or chemical means. And then the FDA industry guidance defines disinfection as a process that destroys pathogens and other microorganisms by physical or chemical means. They go on to say that disinfection processes do not ensure the same margin of safety associated with the sterilization process. AAMI TIR 12 tells us that manufacturers must perform product testing to validate the ability of a device to be successfully disinfected. They also show that the rinsing procedure must be adequate to remove residual disinfectant after disinfection is achieved. FDA guidance tells us that cleaning should be validated separately from disinfection or sterilization, and it says that the ability of the disinfection method to successfully reprocess the device features should be validated. Now, when we talk about disinfection, there are essentially 3 levels of disinfection, and these are all based on the Spalding classification system, which has been around longer than I have. There is low-level disinfection, intermediate-level disinfection, and high-level disinfection. Low-level disinfection is is usually effective in killing vegetative organisms, bacteria, fungi, and lipid viruses. It cannot be relied on to destroy mycobacteria nor spores, and it doesn't work very well against non-lipid viruses. Low-level disinfection is typically used for non-critical medical devices or items that contact intact patient skin. Intermediate level disinfection will kill viruses, it will kill some mycobacteria, and is efficacious against fungi and, of course, vegetative bacteria. It cannot be relied on to destroy bacterial spores. Intermediate level disinfection is typically used for medical equipment surfaces likely to become contaminated by indirect patient contact, usually with procedures involving mucous membrane contact. High-level disinfection, it kills microorganisms, but does not necessarily kill large numbers of bacterial spores. It is typically used for semi-critical and critical medical devices unable to be sterilized, which contact mucous membranes or non-intact skin, or for that matter, any other sterile part of the body, if it cannot be sterilized. And then there are essentially two methods of disinfection. You have thermal disinfection or chemical disinfection. Thermal disinfection is typically done by the action of moist heat in a washer disinfector. Process temperatures usually range from 82 to 95 degrees Celsius. It's used primarily as an interim disinfection step, and this is done to protect healthcare workers. So you can remember back to the diagram where we looked at the processing cycle. The interim disinfection step occurs immediately prior to the inspection and packaging step. So that inspection and packaging work is often done in an environment where there's lesser PPE, personal protective equipment. Often the workers who are doing that work do not have gloved hands. They're not going to have face shields or goggles or anything. So we do want to make sure that devices, after contact with the patient and after a procedure, are safe for them to be handling during their inspection, assembly, packaging work, whatever they're doing prior to sterilization. So thermal disinfection in a washer disinfector is usually considered an interim disinfection step. However, it can be a terminal disinfection step as well. There are some devices that can go through a washer disinfector and having done so are immediately ready for patient use in the next procedure. Chemical disinfection are usually done in an AER, which is an automated endoscope reprocessor, or they're done with like a chemical soak in a sink or a bucket, a pail, a pan, whatever. It's achieved by the action of one or more chemicals. It's used primarily as a terminal disinfection step to protect patients, but it can also be an interim disinfection step in some circumstances. So for instance, if you have a pop-up wipe disinfectant, you might want to be able to use that as an interim cleaning process prior to doing any kind of intricate inspection or assembly or packaging of a device before it gets sterilized. Each unique combination of disinfection processes and methods should be validated separately. So for instance, if in your IFU, you recommend an intermediate level thermal disinfection in a washer disinfector, and then you want a terminal high level chemical disinfection because your device for whatever reason can't be sterilized, you should be validating both of those processes separately. So let's look at validation of the disinfection process. I got to tell you, when I first started preparing these slides, I tried to get the entire disinfection process validation onto a single slide, and I couldn't do it. So I bumped it to 2, and I actually ended up going to 3. So I broke this down into 3 phases. The first step that we do during a disinfection process validation is we determine recovery efficiency. So like cleaning, we start with 6 replicate devices. And these are all going to be control devices. They're not actually the test devices. We then go on and we prepare a low-level organism suspension and separately a high-level organism suspension. And while we're talking about organisms, I should be clear that when we are validating low-level, intermediate-level, or high-level disinfection, there are different acceptance criteria for each of these. So for low-level disinfection, the current acceptance criteria is a 6-log reduction of these 4 organisms. There's some variability in the Klebsiella, but generally the first 3 are clearly defined in the technical information reports. If we're doing intermediate level, they use those same 4 organisms, and then we add a mycobacterium strain. So at STERIS, if we're validating a chemical disinfection process like with a pop-up wipe, we use Mycobacterium terreae, or if we're validating a thermal disinfection process in a washer disinfector, we actually recommend using Mycobacterium hesiacum because it's a thermophile. It's actually harder to kill in a washer disinfector. So for low level, we have a 6-log reduction of these 4 organisms. For intermediate level, we still need to show that 6-log reduction of those 4 organisms, but we need to show a 3-log reduction of the Mycobacteria organism. For high-level disinfection, we only test the Mycobacterium species, and then we have to show a 6-log reduction for high-level disinfection. So those are the acceptance criteria. So we prepare a low-level organism suspension for one of these organisms and a high-level suspension for the same organism. And then we enumerate both of these suspensions so we know, how many organisms per microliter we're starting with. Then we inoculate 3 low-level recovery efficiency devices and 3 high-level recovery efficiency devices. And I'll walk through a little bit later why we do both low-level and high-level. We'll get to that in a bit. I'm not going to discuss that right now. But we do both of them, inoculated low levels, inoculated high level. And then we extract and enumerate the low-level recovery devices and the high-level recovery devices. And then we calculate our recovery efficiency by comparing the known quantity of organisms that we put on the device versus the recovered organism that we got from the device. So doing that math gives us the percentage of recovery efficiency that we have, and conversely, a correction factor can be determined. We do the same thing with a high-level recovery, where we determine a high-level recovery efficiency. They're not going to be the same. Often devices in vastly different numbers have a different percentage of recovery. So we do high-level and low-level recovery efficiencies. And then these devices are repurposed for the next phase, and this high-level organism suspension is also used again later on. So let's talk about the second phase of a disinfection process, which is the neutralizer verification. Now, when we perform a chemical disinfection process, we have to be able to stop the microbicidal effect of the disinfectant at a specific point in time. So this requires the use of a neutralizer. And when we're using a neutralizer, we need to be sure that whatever that neutralizer is, that it doesn't interfere with the test method other than neutralizing the effect of the disinfectant. So we want to make sure that that neutralizer doesn't also kill organisms, just like the disinfectant is. So again, we start with 6 replica control devices. These are the same 6 that we started with for the recovery efficiency. They're being repurposed for this phase of the study. We perform minimal disinfection on 3 of these devices. Then we assign those 3 devices to what we call the test group, and we assign the other 3 devices to a toxicity group. And then we prepare what we call a very low-level organism suspension, which is going to be even fewer organisms per microliter than what we use for the low-level recovery efficiency. So we call it a very low-level organism suspension. We enumerate that to determine how many organisms per microliter we have. And then we add extract fluid to an extraction vessel that we call the viability group. So we're introducing a 3rd group. We add extract fluid to the toxicity group. And then we add neutralizer, whatever we're using as the neutralizer, to the test group. And then we inoculate each one of these vessels, we don't actually inoculate the devices, we inoculate the solution inside the vessels with known quantities of the very low level suspension. Then we extract and enumerate recoverable microorganisms from each one of these three categories. And then we determine neutralizer efficacy by looking at the values from the toxicity group versus the test group. And this assures us that the neutralization method was effective in inhibiting or stopping the antimicrobial properties of the disinfectant. And then we determine neutralizer toxicity by comparing the viability group to the toxicity group. And this ensures that the neutralization method does not impair the recovery of viable microorganisms. And then, of course, devices are repurposed for the next phase, which we're going to go on to slide 3. So this is the actual process validation. Now that we've done all the preliminary work, we know what our recovery efficiencies are, we know that our neutralizer works just fine. Now we can actually validate the process. So we'll start with 3 control devices and 3 test devices. We use that same high-level organism suspension that we use on the first of 3 slides during the high-level recovery efficiency, and we inoculate positive control devices and we inoculate test devices. After we inoculate the test devices, we perform a minimal disinfection process. So again, this is shortest contact time, lowest concentration level of the disinfectant. So it's a minimal disinfection process. And then we extract and enumerate both the test devices and the positive device controls. Now remember, the positive device controls are not disinfected. So after we extract both of those and we get values from those, we apply the high-level correction factor to these devices that were not disinfected, and we use the high-level correction factor because we expect a large number of organisms to be on these devices. And then we apply the low-level correction factor to the test devices because these have been disinfected, so we expect to see low levels of organisms on these. So we apply the correction factors and then determine what kind of log reduction we have for that device. So we take the value from our positive device control and we subtract the value from the test device and demonstrate either a 3 log or a 6 log reduction, depending on what our acceptance criteria is. And then guess what? We have to repeat this whole process for the next organism. Once all organisms, for instance, if we're doing intermediate level disinfection, we repeat this whole process 5 times for each one of the 5 different organisms. So after we've tested all 5 of the organisms, we disinfect 3 test devices again with a 2x maximal disinfection process. And then again, we will do cytotoxicity testing on these to demonstrate that there's no toxic levels of residual chemistries on the device after multiple uses. So for those of you who actually want to see the entire process in one picture, this is what it looks like. So up here we have the recovery efficiency section. This is the neutralization section. And then down here we have the actual process validation. And then we loop back, start over again, and do the whole process again, loop back, and we do that for each one of the organisms. So high-level disinfection is really easy. We only have to go through it one time because we're only testing the one organism. But you can imagine there's a lot of moving parts to this puzzle when we're validating a low-level or an intermediate-level disinfection process. I want to take a really quick minute here to talk about the differences between disinfection and sterilization. So from the FDA point of view, disinfection and sterilization are different types of terminal processes. So you can see this is from their diagram that's presented in their industry guidance. It says you use the device, point of use processing, cleaning, and then either disinfection or sterilization. So this is a very common presentation from the FDA perspective. Their guidance document essentially does not address interim disinfection at all. So they review disinfection and sterilization as alternate forms of a single required microbicidal process. So all throughout their document, you can see disinfect or sterilize, disinfect or clean, clean and disinfect or clean and sterilize, clean and disinfected or clean and sterilized, or. It's all throughout their document. This isn't all of them. There are many, many more. So they essentially say that they don't address interim disinfection at all. The international community, however, does somewhat recognize two distinct disinfection processes, and it's becoming more and more widely recognized and widely understood. Back in 2010, when TIR 12 was published, they actually said that thorough cleaning, rinsing, and drying, and when necessary, additional microbicidal processes to ensure personnel activity. Now, this step occurs before sterilization. So this is essentially interim disinfection. 17664, 2017 lays out, you have pre-treatment, preparation before cleaning, disinfection, and then down here you have sterilization. So you can see step D is an interim disinfection process. I would really like to have seen H say either disinfection or sterilization, because not all devices are able to be sterilized. But at least I like to see this distinction of a disinfection process that's up here right after cleaning. And then throughout ISO 17664, you can see disinfected and or sterilized and or, and it goes all through the document this way. So you can see that there is becoming a recognition of this term, this aspect of interim disinfection, which is fairly new to the industry. So let's talk about sterilization. Now that we've done cleaning and disinfection, sterilization is kind of the easiest of the three. Sterilization definitions with ISO 11139 and TIR 12 and TIR 30 all share the same definition. It's a validated process used to render product free from viable microorganisms. ISO 17664 says it's a process to render product free from viable microorganisms. And FDA industry guidance says disinfection and sterilization processes are intended to kill microorganisms. Validated processes are used to render product free from viable microorganisms. That's specifically sterilization, not disinfection. TIR 12 tells us that manufacturers of reusable medical devices intended to be sterile have the responsibility to support label claims by providing complete, validated, and comprehensive written instructions. The FDA guidance says that the method of cleaning or disinfection needs to be validated, microbicidal method for disinfection or sterilization. They also say that drying needs to be validated. So if you have a steam sterilization process, you want to validate that drying process. And then they go on to say that you validate the sterilization cycle using specifications that are consistent with conventional parameters. Include a sterilization process that you have attained a sterility assurance level of 10 to the minus 6 or 10 to the minus 3 as appropriate. Validation data should be generated in FDA-cleared sterilizers and with FDA-cleared sterilization accessories. There are essentially two categories of steam sterilization. You have dynamic air removal and you have gravity displacement. Dynamic air removal is considered a pre-vacuum process or a steam flush pressure pulse. Some of you have heard the term SFPP. Both of those are considered dynamic air removal processes in that they use an active, aggressive method to pull air out of the chamber, while gravity displacement relies exclusively on gravity and flushing air out the bottom of a chamber. We also have VHP sterilization, which there are several types of VHP processes. In the STERIS vPro, we have the fast non-lumen, the non-lumen, the flexible, and the lumen. And then the ASP STERAD devices have an express cycle, a standard cycle, a flex cycle, and a dual cycle. There are several steam sterilization processes, and you can program a sterilizer to do just about anything if you have a good sterilizer. But there are certain processes that are widely recognized, especially in the US under FDA requirements. And these guidances are defined in AMI ST79. They're in TIR 12. They're in the FDA industry guidance. I think they're also in a fourth. It might be ST88 or something. I don't remember. It's in a fourth guidance document also, where it defines these specific processes for reusable devices. And FDA really requires device manufacturers to validate one of these methods to be used domestically. So we have 4 minutes at 132 degrees C. That's probably the most widely used process domestically. So you can see here 32 minutes for 4 minutes in the Western Hemisphere. In Europe, they prefer a 134 degrees C process for 3 minutes. So just like cleaning and disinfection, each unique process needs to be validated separately. So if you plan to market your devices in the US and in Europe, you'll want to validate both of these pre-vacuum processes, and you're going to be looking at 2 sterilization process validations. And if you want to validate a VHP process, you're looking at a 3rd validation for the repro and a 4th validation for the STERIS, or any combination of the above. So let's look at how we validate a sterilization process. We start with the devices or the tray system to be validated. Often with reusable devices, we're looking at something like an orthopedic tray that has many devices in it. So we call it a tray system. We perform a microbiological performance qualification. By doing this, we place microbial challenges throughout the tray, or if it's a singular device, we'll put multiple devices inside a sterile barrier system, and we'll seed those sterile barrier systems with microbial challenges. We use a sterile barrier system, as mentioned. We execute half-cycle runs, usually, for steam sterilization. Then we remove the process items. We allow them to cool. We culture the BIs. We confirm that none of them have any viable growth. And then we extrapolate from that value a full cycle sterility assurance level that's going to be less than or equal to 10 to the minus 6th. After we've executed the microbiological performance qualification, we do what's called a physical performance qualification. So this culture microbial challenges phase, this takes 7 days. This one step takes 7 days. So while those BIs are being tested for those 7 days, we actually go on to perform the physical performance qualification during that window of opportunity, so we're not losing time. So we'll start by placing temperature sensors throughout the system, whether it's going to be in the tray, or if it's a single device in a pouch, we'll put temperature sensors inside multiple devices in multiple pouches. We use a sterile barrier system, as mentioned. We execute full cycle PPQ runs. We use half cycles for the MPQ. We use full cycles for PPQ. And then we remove the process items and allow them to cool. We assess for residual moisture. Here's where we validate the dry time that I mentioned on the previous slide. We confirm that the process temperature was achieved. So if you're running a 132 degrees C process, you want to confirm that each one of the thermocouples that you placed throughout your system all achieved that sterilization temperature. And then we determine a full cycle S sub 0, which is greater than or equal to 12 minutes. So one more thing, I wanted to talk a little bit about some guidance documents that are coming up. ISO 17664-2 is going to be published very soon. So the 17664 document deals primarily with patient contacting devices. This -2 document is for medical devices not intended for direct patient contact. So this guidance document is something that we've been waiting for a long time to see, because both 17664, the original one, and TIR 12, they both exclude medical equipment. So if you have a console sitting on a cart or something that has no direct patient contact, those standards exclude those types of devices. So we have no guidance for what do we need to do to make sure that I can effectively clean and disinfect that product if I have no guidance or standards to use. So this is a much needed document. I'm really glad to see this coming down the pipe. There's also ST98, which is going to supersede TIR 30, which I mentioned earlier. TIR 30 is the one that deals with a compendium of cleaning processes and acceptance criteria. This is upgraded to a standard. It goes from a TIR, which is a technical information report, it's being upgraded to ST, which is a standard, which carries the weight of law. So it's going to be a really good introduction. It will work very closely in parallel with the FDA industry guidance to clarify and define test methods for cleaning process validations. And I believe the standard is going to present clear and well-defined acceptance criteria, which is going to be very nice to have to reference when we're validating cleaning processes. And then TIR 12 has also recently undergone revision and will be published very soon. This updated version has some very specific verbiage in it regarding the differences between interim disinfection and terminal disinfection, which I'm glad to see. A lot of the stuff in TIR 12 that used to deal with cleaning processes has been vastly reduced because they were aware that ST98 was coming, so they didn't want to duplicate the work or contradict anything that ST98 says. So a lot of the stuff in TIR 12 that used to deal with cleaning has been pulled out, and it's now dealing primarily with disinfection and sterilization.
Thanks again for joining us today, and this concludes the webinar.